And now I call upon Dr. Santosh Gopi Krishna. He will be sharing his new insights into DME and DR management with biomarkers.
So Dr. Santosh is a consultant at Naranetralaya in the Department of Vitreoretina, and he has a lot of work on biomarkers. So looking forward to hear what you have to say.
Thank you. Morning. Thank you, Dr. Pish and Dr. AOS for this opportunity. So I'll be, it's a basic sciences talk. I'll try to kind of make it simple. It's basically new insights into the biomarkers, especially I'll be talking about molecular biomarkers, the soluble factors, not the imaging and other biomarkers. So to start with, we need to understand what exactly is a biomarker. Biomarker is a characteristic feature or finding that is measurable, which is predictive of outcome, and it aids in prognosis of the therapeutic response. It's a valuable tool for detecting the early stages of the disease, tracking progression, and monitoring treatment response. So even the needs and the requirements of the biomarkers also have changed over a period of time. You can see that the prognosis is most meaningful if the level is clinically actionable, and biomonitoring is also included in the newer definitions of the biomarkers. So we all know this, what exactly causes diabetic retinopathy and diabetic macular edema: chronic hyperglycemia, oxidative stress, and it starts. Hypoxia, ischemia, and blood retinal barrier breakdown. It could be either neovascular retinopathy or it could be diabetic macular edema, which is a vision threatening. So this is again a slide showing what really happens to the blood retinal barrier. You can see the oxidative stress at the baseline here, and which starts and causes leakage. You can see the blood retinal barrier breakdown. There could be neovascularization that happens. Ang2 and Ang1, these are the factors that act against each other, and VEGF is the main factor that we know and which has been targeted time and again. This again is a picture that kind of depicts what really happens is in a normal retina and what happens in a diabetic retina, and especially in the endothelial cells. You can see what really goes wrong. There can be immune cell activation, there can be gliosis, there is damage to the vascular basement membrane, altered endothelial mural cell interactions, and especially the impaired neurovascular coupling, which leads to neuropathy. And again, this is again important, not just the inflammatory factors, even the angiogenic factors. There is a complex interplay and balance that needs to be achieved. Once there's a tilt, we obviously know that it goes in for an angiogenesis. In healthy conditions, these are the cytokines, chemokines, and adhesion molecules which are normal. But in diabetic retinopathy, everything shoots up. And the balance is what is important, which kind of leads to vision-threatening diabetic retinopathy. So there are several studies and meta-analysis which have shown that these are the potential ocular biomarkers for diabetic retinopathy. Several others are there, but these are the important ones. And these are potential systemic biomarkers for diabetic retinopathy. So we can check these biomarkers even in blood as well as aqueous and vitreous as well as in TS. So this is a slide showing the different inflammatory cytokines, the chemokines, adhesion molecules, growth factors. So you can see what all happens in different stages of diabetic retinopathy. You can see the progression. Some are linked to the NPDR and increased with DR progression, but the basic things are the TNF-alpha and the interleukins that starts the process, and from there it picks up and it goes into a complex cascade. So these are the different mechanism-based treatments. So when you give a steroid, everything kind of comes down, all the inflammatory biomarkers, even the VEGF to some extent. But when you look at the TNF-alpha and all, these are the molecules that are being explored. Some are kind of terminated because of there was not proper response, and some the research is still on. So if you look at previous papers, they showed that inflammatory biomarkers like IL-6 and VEGFs are obviously elevated. And when you have an ICAM, which is there earlier, it gives a protective role. So that is what the earlier papers have told. Now, if you see this paper, it shows that not only VEGF, we need to look beyond VEGF. So there is an extra VEGF cascade that happens, and which needs to be targeted for better treatment. So even the ICAM molecules correlates with the disease severity, especially in recurring macular edema. So this is an old paper which shows IL-6 and IL-8 are increased in different types of macular edema. You can see a cystoid or a mixed macular edema, and these two things are elevated. So this is work done by Raju Raman sir. So they have found that IL-1 RA is elevated, and you could see the potential increase in this biomarker over the severity of diabetic retinopathy. Then again, soluble CD4 counts in aqueous humor are elevated when you see these HRDs in diabetic macular edema. And these are the newer papers which again reiterate the same thing, saying that these biomarkers are extremely important and these need to be targeted. You can see these 6 biomarkers that are mentioned here, including VEGF, ICAM and all, which kind of play an important role in diabetic macular edema and diabetic retinopathy. So these are the molecular signatures and approved agents. If you look at across neovascular AMD, DME, PDR and RVO, so these are the molecular signatures that can be targeted, and these are the FDA approved agents across these diseases. Angiogenesis is an important part. There are multiple targets that could be achieved, but it's not necessarily that they'll kind of reach a viable endpoint. So it's a different ball game, and it's difficult to kind of get a potential therapeutic target in such cases. So these are the ones which are approved drugs. These are under research. You can see many are in phase 2, phase 3 studies, and most of them target VEGF, some target Ang2, and there are other molecules also that are being targeted. So ultimately, the reliability and the end product is what is important. So coming to a sneak peek into what's really happening at Narayana Nethralaya. So we actually have this project strategy wherein we look at aqueous humor. We look at the profiling, we see at the severity of diabetic retinopathy, types of diabetic macular edema, and the treatment outcomes, especially if it's a treatment naive case or recurring or non-responding diabetic macular edema. And then we even check these samples across in comparison with the controls as well as in PDR cases, especially vitreous hemorrhage. And then it comes to in vitro trials, wherein we kind of see these factors and check how they react with other molecules, anti-VEGFs and TNF and all. So these are on RPE cell lines or HUVEC cell lines, and in vivo, especially the mice models. So this is the samples type and strategy, how we go about. We collect these and they're stored at -80, and CB analysis is done. Around 50 changed biomarkers are usually done. And there was a positive association that was established long back in our lab between the aqueous as well as vitreous. And if you look at different types of macular edema, you can see the various factors are there, but these are little difficult to interpret and kind of come to a conclusion because statistical analysis is not very easy. There are multiple factors involved. So to come out, we can publish a paper saying that these are so and so, but to have a statistical analysis, it's a little extremely important to have large data. This is again across severity of diabetic retinopathy, non-severe DR, severe DR, and PDR. You can see the complex interplay of these factors again. Now, when it comes to vitreous, you can see a clear change in the biomarkers when it compares to controls. When you have a vitreous hemorrhage and PDR, all these factors are definitely elevated. And again, these are the interplay between aqueous as well as vitreous humors in PDR and normal control cases. ICAM is one molecule which we really kind of found interesting. That is because it's an adhesion molecule, and we have found that it is extremely important in non-responding DME, and somehow like we are proceeding in this lines of finding a more substantial kind of research on this. Then coming to in vitro, so we have these RPE cell lines. You can see the mRNA expression of the ICAM and IL-6. When you have a TNF alpha inflammatory response, the ICAM goes high, even IL-6 goes high. So the mRNA expression and secretions, even later also after 24 hours, it remains high. Then again, this is the same kind of a thing. It shows that inflammatory stimulus induces secretion of ICAM, which is independent of VEGF. So when you kind of subject these RPE cell lines or HUVEC cell lines to inflammatory stimulus like TNF alpha, especially the ICAM goes high, whereas the VEGF remains low. So it definitely means that there is an alternate pathway that is responsible for diabetic retinopathy and diabetic macular edema. This is again a similar kind of thing wherein if you have a increased TNF alpha, you can see fourfold inflammatory stimulus in these conditions. And then again, 72 post, especially if you subject these cells to hyperglycemic stress, then again the increase in ICAM is several fold, and you can see the changes in ICAM as well. So this is again kind of reiterating the importance of ICAM when you subject the patients with different combinations of TNF and you've kind of given VEGF at anti-VEGF and TNF with anti-VEGF. This is how the molecules and the solutes behave. It's the same reiteration, and these are some papers what we have published over a period of time. You can see this S100 and these are the molecules which kind of say that the damage related to molecular proteins, they directly correlate to the severity of diabetic retinopathy. As the severity of diabetic retinopathy increases, these moleculars are found in excess. This is again a different lens protein that is expressed in astrocytes. So when the expression is more, especially in PDR, if you see, there is a lower level of these astrocytes in vitreous. So decorin is a small leucine-rich proteoglycan. It's a part of retinal endothelial ECM. These elevated levels are seen in severe DR groups again. STING, this is a newer paper what we published. When it's, this STING is a stimulator of interferon genes. It regulates cell senescence and inflammation, and it's extremely important for the development and progression of DR. And in mice models, we have seen that targeting STINGs could be an effective treatment strategy for early DR. So this is the newest paper wherein we have seen microRNA from RP cell line. It regulates multiple gene products. So it has an important role in glycolysis and angiogenesis. So RP cells, when they are under high glucose stress, the mRNA is elevated, and this kind of alters the glycolytic pathways, and increased expression of VEGF is noted. So coming to our newest thing, the biomarker pathfinder, we have 8 molecules that could be identified. It's a readout that comes in 2 to 4 hours. So this is how it's done, and the TRs is the collected, they put in a buffer, and you can put it in the cartridge here. And once you put it in the machine, you get a readout after 2 to 4 hours, depending on the number of molecules that we read. So we try to look into whether AI can help to understand the complex interplay of these biomarkers. So we had done these 8 biomarkers, and we have taken a cohort of 192 controls and 175 treatment eyes. So what we have seen is there was a good correlation, and the sensitivity and specificity was really good. What AI had found was these 3 biomarkers which were really important, the ICAM, MMP9 and VEGF-A. In some cohorts which didn't have a diabetic retinopathy, we found that the VEGF-A was high, and these we named it as vascular disease suspects.
Santosh, you have to conclude.
Yeah. So the vascular disease suspects were more prone to kind of going for a diabetic retinopathy and a vision-threatening diabetic retinopathy. I'll just skip these. So VAS study is the newest one Dr. Chetra had presented recently as a free paper. This is a relationship between immune factors between vitreous, aqueous serum, and tears. The cluster, if you can see, the cluster 2 has more molecules, and you can say that the correlation is better between vitreous and aqueous. So it definitely means that aqueous can be used as a substitute for vitreous to measure these biomarkers. So to conclude, these are the molecules that are really being considered, anti-angiogenesis, even RAS blockage are being considered. So DR and DME, there's a complex interplay of soluble factors. Pathways are beyond VEGF. R&D, especially once we start targeting these molecules, there could be a personalized treatment, especially like molecules like faricimab. So to conclude, it would be right to say that the ideal biomarker is the one through which the disease comes about, or through which an intervention alters the disease. Thank you so much.
Thank you. Thank you, Dr. Santosh. I think it was a real deep insight into the biomarkers, but I think what's the clinical application as of today?
As of today, we have been finding different pathways of initial starting of diabetic retinopathy. The molecules, I mean, especially the cascade which can start diabetic retinopathy. I know it's like, I mean, when it translates to translational science, especially the medicine, it's difficult, it's a long way to go. But these are the pathways which could be targeted maybe in the near futures.
Yeah, wonderful presentation and wonderful work you're doing. Thank you.